Okay, we can start. Today we have the great privilege of having with us as a speaker Pietro Invernizzi. Pietro is not only a good friend of all of us, but he's also an outstanding scientist in autoimmune hepatic disease, and in particular the genetic aspect of autoimmune hepatic disease. Pietro was the chair or the conductor, let's put it this way, of a large group scattered around the world, who actually collected a lot of different cases of PBC and other diseases related to autoimmune, and actually published a very landmark paper on this in Gastroenterology, I think 5 years ago. And this was followed by other studies, and altogether this study paved the way to reshape from the scratch all our current views on the involvement of genetic environment in autoimmune hepatic disease. Pietro got the time and attention to accept our invitation, although his time is so busy doing the director of the largest department of medicine in Italy, and we are supporting him very much on this endeavor. But today he switched from the director role to the science role, and so I give the podium, the virtual podium, to Pietro. Please go ahead, Pietro. Thank you very much for being with us.
Thank you, Claudio, for the very kind and presentation, introduction. Very kind, too much. And I'm very proud and glad to be your friend.
That's a real truth.
Community, of your community. So my task today will be to talk to you about genetics. I was asked to talk about genetics of autoimmune liver diseases, but because just before the COVID, I don't remember exactly when, I came and in person and gave a lecture on PBC, and I talked to you a lot about genetics in PBC. So I try today to just quote some information on autoimmune liver diseases, and but with the main goal to give you an overview and try to give you the what they think about the novel trajectory on the field. Okay, so let me share my slide. Okay. Okay, here we are. Can you see?
Yes.
Yes.
Okay, perfect. Okay. So, and my goal today is to tell you what we have learned up to today from GWAS, Genome-Wide Association Study in Autoimmune Liver Diseases. Trying to say what the benefit and the limitations of GWAS, and as I said, to share with you the novel trajectory. We have to investigate genetic architecture of autoimmune liver diseases. That means autoimmune diseases or genetically complex diseases. So let's start with what we have learned from GWAS, not going in details on this, but starting from definition, we have to say that between Mendelian or monogenic disorders based on defect in rare variants and polygenic and complex disorders due to variants or with the role of common variants in the development and progression of disease, we are talking about autoimmune liver diseases. We are on the group of polygenic and complex disorders. That is disorder with sorry, I'm trying to move if I can, something like, okay, like that. So the disorder where many genetic variants of low risk interact with environmental factors, so it's quite complex. let's say that this is the way that most diseases are heritable. So in talking about autoimmune liver diseases, we might divide what we know about the genetic architecture of these diseases into different parts or era. The pre-GWAS era that finished when the Human Genome Project was finished, was completed in 2008, 2010, more or less, and the GWAS era from that time forward, until 2008, the most of the association, genetic association, I thought many genetic variants were reported, but the strongest one and the more consistent evidences on the architecture, on genetic architecture, were related to HLA variants. Mainly, here I'm writing down the one associated with PBC, but HLA was also for PSC, primary sclerosing cholangitis, and autoimmune hepatitis, the main and strongest association. So, and I'm basically talking about the classical the study relating to dealing with single nucleotide polymorphism, and the classical study that try to find difference between patient and a healthy subject with evidences of association or no association. And from the very beginning, we had, after the Human Genome Project was completed, a so-called first wave of GWAS, focusing on common variants. And so we had also for PBC and for any autoimmune liver diseases, a number of studies reporting association, reporting loci or genes associated with the disease. And here, a summary of the loci, found to be associated with PBC from the very early studies in 2009, 2010, based on North American and Italian and North American series. And then other studies were came later from UK, Japan, now also from China. But here a list that is not the world list, but you can see that actually I want to let you see that most of these loci are coding for genes, immune-related genes. And the second comment I want to make is that in any studies up to date, the consistent association and the strongest association is with the HLA region in the chromosome 6. After this first wave of GWAS, then later on other more a so-called second wave, focusing and going more in deep in association and try to better characterize the architecture is still ongoing. We are working on this, but later in my talk I will give you more details. And talking about PBC and what we know on genetics in PBC, let's say that from our group, mainly from our group, but also from other groups, there has been a number of evidences showing a major genetic defect in sex chromosome. The strongest and the first evidence and the strongest one was the demonstration of an enhanced X monosomy in female with PBC, and that was that it has been described as preferential, as due to a preferential X chromosome loss. We still don't know if the X chromosome from paternal or with the paternal origin or maternal, but one of the two is preferentially lost. And then also it has been reported an increased Y chromosome loss in male with PBC compared with healthy male. So overall, all this kind of evidences bring us to hypothesize that a sort of haploinsufficiency for some key immune-related genes and proteins play a role in masking PBC susceptibility genes, and so play a role in the pathogenesis of the disease. And this is what we know on PBC up to now, up to date. Then moving to PSC, what we know, a similar story. I'm not going in detail, but here a summary from Zhang and Carlsen, Tom Carlsen, published a few years ago, just summarizing a number of loci and genes found to be associated with the disease, with the high, strongest association again in AIH, HLA, sorry, HLA. And here again, from the same reviews, what we the effort to understand if this general what we know about the architecture, genetic architecture in PSC is similar with other immune-related or autoimmune diseases, and actually it is clear that a similar genetic architecture or association are found in a number of autoimmune disorders. So just suggesting a common trait for these complex diseases. In particular, most of up to half of the genes or loci associated with PSC are shared with IBD, inflammatory bowel disease, ulcerative colitis, or Crohn's disease. While even higher similarity was found with celiac disease, or I remember from the data the drug-induced liver diseases. So this is quite important for thinking about our knowledge of autoimmunity in general. And again, HLA, as you can see here clearly around the chromosome 6, is the strongest association, with few exceptions, is the strongest genetic association in any or most of the autoimmune diseases. So still in the PSC, looking at the molecules found to be associated with a genetic association, it clearly recapitulates the pathogenesis. In fact, if here in this cartoon, you see a number of candidate loci that were identified in T cells in PSC, but also some loci associated in cholangiocyte and stromal cells with a key role, cells with a key role in fibrogenesis or the fibrogenetic processes. And so I'm not going in detail, but this is quite suggestive and intriguing for PSC, for PBC and PSC, and what we know in about the pathogenesis of this disease. So very scanty data compared to PBC and PSC. We have scanty information on genetic in autoimmune hepatitis. Here the study published a few years ago in Gastroenterology 2014 by in Holland, a group from Holland, showing only one strong association with HLA. And I couldn't show you. I shared with you because it's impressed, but not yet in available, publicly available. We have similar data with some new novel association from a very nice study from on genetics or GWAS study from Chinese AIH patients. So, but you will see soon, and I can anticipate that we have similar results. So, but now, and what? Now that we know all this, now that we have all this information, and now what? And so my seminar now will deal with what we should do now, the challenges we have. And as I already said to you, the community is focusing now more and more on rare variants, not only common variants, and on using dense arrays, on performing DNA sequencing, RNA sequencing, starting to study more other population, ethnic population, and so on and so on. So here the more general part of my seminar. So the benefit, relating on benefit and limitation of GWAS. And so talking about GWAS in this cartoon, let's say that GWAS start with the goal of the GWAS is to find association between genetic trait and clinical trait. So first, we need to identify the proper population, controls and cases. Then we have a number of genetic approach, and in this case, we use the SNP array and imputation, and we are starting to use whole genome sequencing more and more. We have a number of statistical tests to be used, and the real challenge nowadays is to perform study of linkage disequilibrium with the aim of identify the functional characterization of this association. And here the number of possible approaches we are dealing with. So trying to study the chromatin accessibility, the histone modification, DNA modification, so the gene expression system controls, and so on. And of course, finally, we are since a while trying to validate result experimentally, so directly with cell lines or a number of methodological approaches. And here the challenges in interpretation of GWAS association. So we of course it's important to find association, but we need to know much more. We want to know much more. So we want to answer these questions. Which are the causal variants? Not only, on the top of this cartoon, you see on the left side the Manhattan plot, but then we need to move, as in the on the right side of the panel, we need to focus on single example locus. This is an example locus. So to focus on a locus and going more in deep, but trying to identify the causal variants And an approach is a fine mapping, as I already said. We really would want to identify in which cell types do the variants act. And these are an approach is in SNPs enrichment. And I'm going to talk to you in a while. And then again, and then also try to know which genes are regulated by the variants. And again, there are some approaches as the co-localization that more and more groups are now trying to work on. So, and to date, what we can say about the discoveries that can be made by GWAS. So it's like in this cartoon, we can think as an iceberg. So the top of the iceberg, out of the water. Here, the discovery in the cartoon, you can read the discovery we can today perform. So we can study. We have data on European population mainly, and we can work at additive model in GWAS analysis, and easy to measure phenotypes when we have a clear phenotype. But below the water, some example of what we really want to do, we are starting to do, and we have to do, and in the future. So we have to develop different study design, and so I'm not working on larger and more ethnically diverse reference panel, extremely large population, and so on and so on. I'm not going in detail. And here in the same very nice review I suggest you to read from TAM in 2019, a list of benefit and limitations of GWAS, what we can now do or in the near future, and what and the number of limitation we still have on GWAS approaches. I'm not going in detail. So, but starting from what kind of data we have today, and the example of what we can do better. Here in this review, again in a couple of years ago, that summarized how many data we have from genetic data we have from European population compared to population from other geographical area, and is incredible, is amazing how most of the data today available, also from autoimmune liver diseases, are from European populations. And here a very nice example of what we missed performing our study only on European population. So in this study, the authors analyzed, tried to analyze a number of clinical traits, and you see on the left, and the association reported in a number of studies, the GWAS studies, including European, in this case, performing this analysis, very large analysis, actually analysis on a very large data set, including all the data available for non-European ancestry or populations. And it's clearly they found a number of novel associations only because they analyzed adding in the analysis non-European population, the orange bars. compared to the already known association and the replicated one. And as I said, also in autoimmune liver diseases, we are expanding our knowledge, and other groups around the world are starting to perform GWAS on their populations. And here the example of very nice GWAS performed in PBC series in Chinese PBC series. And it's quite interesting to see that other than the already known associations, they found a number. Here you see the CD58 and so interleukin 21 and so on, that were novel, that were not reported in previous GWAS from European series or Caucasian series. So this effort to include all the available data and this kind of in a number of scientific community is underway. And also in PBC, a group of experts with available GWAS data worked together, and last year published these meta-analysis studies, including 10,000 more PBC cases and 20,000 more control case, healthy subject. And in this and actually trying to define with a number of association confirmed by means of this approach, and a number 20 in particular novel loci identified thanks to this kind of approach. And here the Manhattan plot, actually not 20, but 21 additional risk loci, thanks to this approach of meta-analysis as combining based on a combined data set with all the available GWAS data worldwide. So I already mentioned, but saying some word more, going more in deep on whole genome sequencing, and what whole genome sequencing approach can give us in our attempt to understand the pathogenesis or the genetic architecture of rare diseases. And here a study published in last 2020, where a number of subjects, a large number of subjects, were screened by whole genome sequencing. And I'm not going detail, but is an example, a good example actually, of what we showing that we can identify a number of genetic architecture, a number of genes, and that allow basically to perform a molecular diagnosis and identify the causative genes and etiological variants in many diseases. Here a very good example related to, for example, related to primary immune deficiency, PID. In fact, in this study, they clearly show that using in a cohort or genotyping in a cohort, in a very well defined cohort by means of whole genome sequencing, these authors were able to identify on a number of genetic defects, the orange one here reported, in 20 or more genes that were the genes, the defective causing a number of the same number of PIDs. So thanks to this approach, is really we can identify much better the genetic defect underlying PID and other complex trait, disease complex trait. Okay, let's move to polygenic risk scores, another frontiers of genetic studies or genetic studies. So what are polygenic risk scores? Are the measure to estimate genetic propensity to a trait at the individual level, are based on the sum of the risk alleles identified in GWAS. So having a list of loci found to be associated in by means of GWAS in a specific disease, we can develop a polygenic risk scores. And of course, is a key instrument, as a key role in precision, in terms of precision medicine. The power here is an example of the related to the prevalence of inflammatory bowel disease. It's just an example, and that show the power of identifying subject at high risk using and applying a polygenic risk scores. And we developed a polygenic risk score in our series based on the GWAS data from Italian series, cases and controls. I'm not going in detail on the study design, but we were able to, of course, starting from the list of loci found to be associated with PBC, at least in Caucasian PBC, and we developed these polygenic risk scores. I'm not going in detail, but basically applying this polygenic risk score, we can, for example, screening the relatives of patients with PBC or other autoimmune diseases, autoimmune liver diseases, and more in general, autoimmune diseases. Identifying the one with higher risk score for PBC, and maybe in that, and only on that subset of cases. Of course, in the paper, the first paper we are writing down, we don't have this kind of data or evidences, but in the future, we might screen, and then we might test, for example, AMA or liver function test only in those subset of patients. Okay, let me come back to X chromosome. I already told you that there are clear evidence of major defects in sex chromosome, X chromosome in particular. And I want to start from the original observation we had a few years ago, when being interested in the genetics, but also and having performed the one of the first GWAS in PBC, was quite intriguing to see, actually disappointing, to see that no association were reported in X chromosome on the far right of the Manhattan plot, in any Manhattan plot. Here 3 out of the number of Manhattan plot from the number of GWAS in PBC performed and published. So, but at some point, so, and that was the reason we shift our focus on epigenetics. So we stopped to work on genetics, DNA, but starting to study, and we started years ago to study the control system of gene expression. So that is epigenetics. And but at some point, this guy, Cannon, published in 2015 and suggested and actually explained that the reason, at least one of the reasons, most of the GWAS failed to find any association in XWAS was in the was due to statistical reason. So the GWAS is not able to find association, and they proposed a different analysis or a different way to analyze the genetic data and they named XWAS, not GWAS, but XWAS. And so that from that time we start and we perform a GWAS in PBC using most of the data available at that time. So more than GWAS data from more than 5,000 cases, including from Asia, China, and Japan, and 2 times more controls. And here the study design. So we started performing the XWAS analysis and meta-analysis. We identify a number, 100 more or less, novel loci, and then the main result from this approach, and everything is published now in Gastroenterology last year, was the identification of a super enhancer that is a gene that it clearly controls a number of other genes, and among them FOXP3 that is well known to have a role in the Treg T lymphocyte, T regulatory cells, and or2 D5 involved in the activities and control of T regulatory is the main result from this study. Here some more details, the Manhattan plot and the 102 leads genetic variants we found in 5 cohorts. 2 of the number of genes that were controlled by the super enhancer. I'm not going in detail, the molecular detail, I think I don't have time enough. And FOX04. And here, very intriguing observation we had, that is, most of the genes that we found associated in X chromosome, located in the X chromosome, showed in a number, and that was data, and that were data from our group as well, show a differentially methylated pattern. So in some way, the same genes located at the X chromosome that codes for more than 1,000 genes. So the most of the genes associated in PBC are also the same gene that has been reported. It has been reported having a differentially methylation status. That is quite intriguing. So we should work more on this direction. Here the super enhancer, FOXP3, and here some functional data we perform, or evidences we had, showing that in a number of cases and control, and we isolated circulating cells, and we show a differential expression of these 2 genes in circulating, that is, immune cells from PBC compared to non-PBC. So just showing it's not a real functional demonstration of the role, but it's something, it's showing that there is something to, at least to start a novel study focus on this. Okay, I still have 2 points. I don't know how, Claudio, how much time I still have.
You can keep going, Alex.
Okay. Then we, another frontiers here is the using of the machine learning in artificial intelligence and machine learning revolution. So we, all of us know that the potential of these approaches. Here, the father of machine learning used the word they used to describe machine learning in 1959. He said, Arthur Samuel said, the field of the study that gives computer the ability to learn without being explicitly programmed. So this is the concept of machine learning. And by applying machine learning, we can work on functional genomics. We can work on genotype, phenotype approaches or studies, in gene-gene interaction, in multi-omics integration. That is in many aspects, or many aspects of study or study design. So, and we are working on these, and we are currently analyzing data by using one of the algorithms available. I'm sure you know that nowadays there are a number of algorithms with some pro and contra. We are using Rulex. Alessio Gerussi from our groups is a PhD student, very smart one, but is using this algorithm. And actually, we just published, and it's still in press, not available, but really just published. And this one, this cartoon is the cover of one of the soon issue of Liver International. And we used this approach, and the title of the paper, the studies of the article is Machine Learning in PBC, a novel approach for risk stratification. But in this study, we analyzed a very large data set of clinical information, biochemical information, and so on and so on. So we are using the same algorithm, now analyzing the genetic data, the GWAS data we have. And the plan is to perform some experiment and setting the plan of analysis in the Italian data set, and then to recapitulate or validate in other GWAS setting from Asia or North America and so on. So, but as in this study, based on analyzing the clinical information, we were able, the machine, not we, the machine, the algorithm, was able to, as you can read in few weeks or so, a role, for example, in this study, we show the role of albumin levels in PBC. And this was quite, actually, this was novel. And only the algorithm was able to identify this role for a biochemical parameter. So we are very positive that thanks to this approach, we might find some stronger evidence on some aspect of the genetic architecture. And then finally, the multi-omics integration. Here is the field, the right field for the artificial intelligence and machine learning. We can, of course, mix the use of these approaches, analytical approaches, with the single cells multi-omic and the patient-derived experimental disease models. And so I think this is a very an incredible phase of this scientific story, or we are not facing, living with. Okay, thinking these multi-omics approaches and what we can do with a mix of biological novel approaches, single cells, and the analytical or computational approaches, here a very interesting study that were able to identify a different functional, a different functional role, I would say, of some loci, different variants in specific cells. So they isolate and they perform single cells approaches in different immune cells. You can see here the wall, PBNC, but then CD4, CD8, natural killer, monocyte, and so on. And they check the functional impact of a specific of specific variants in singular loci. So that is amazing, as in my opinion, and opened really opened the possibility to get closer and closer to the etiology, etiopathogenesis of these complex diseases. Okay, here is too much general, I skip, and I move to the very last part of my talk that is related to archaic DNA. This is very fascinating to me. Is a very brand new branch of genetic field, and we are working on that, and we are quite positive we might know much more on the genetic architecture of these diseases, autoimmune liver diseases, but in general. So is basically, I think that I tell you, not in theory, but I tell you what happened. What happened that this guy first, Pabo, Svante Pabo, he's the director, is the father of, still young, but is a father of the field of paleogenetic field. He's the director of the Max Planck in Germany, the leading center dealing with from many aspects, from cultural and to the biological aspect on ancient humans. And he was the first able to sequence the DNA from extincted hominids, Neanderthal, Denisovian, and so on. That happened 10 years ago, more or less. And from them, it was clear that actually they were able to show that we are carrying in our genome a part of genome from these ancient species, Neanderthal, Denisovian, and so on. More or less 1 to 2, 3, 4% of our genome is from these other species. Because many years ago, 50, 60, and years, 1,000 years ago, the species, our species, Homo sapiens, inbred with other species, and so we are still carrying some of their DNA, and this archaic DNA, archaic DNA, it has been shown to be associated with our features, why we are tall or not tall, or we have some features, body features, and also with disease. In 2014, Sriram Sankararanam in Nature showed for the first time an association of the amount of this archaic DNA with some diseases in excess and in more of these and less. He described some genetic desert, especially in X chromosome. And so we started to work on that, and we are currently working with people from the group of Svante, trying to understand if this archaic DNA has a role in autoimmune liver diseases, in PBC. And here the data are coming out, so are not published yet, but from, again, Gerussi is working on this, showing that there are some enrichment in Neanderthal alleles for autoimmune traits, not autoimmune liver disease, but autoimmune in general. I think you know, because that was very fashion, that there are some variant, Neanderthal variant associated with COVID, for example, an infection with by COVID, and the one who published that was Svante Pabo, of course, the same guy I show you. And so I hope that this is not only fascinating, but will help us to understand this disease. Okay, I think I can stop here, and thank you very much for your attention, and open for question, discussion.
Well, thank you very much for this fascinating conference, bringing together science, medicine, paleontology, perspective, artificial intelligence, and you name it. So you are booked for another yellow seminar when the Neanderthal story will be ready, because I'm really fascinated. There is any question from the floor? Don't be shy. If not, let me ask one simple and stupid question. Your data indicate the role of the genes in different diseases. Do you think that at the general practitioner, at the level of the daily basis medicine, this may play a role in preventing or treating or discovering earlier than usual the disorder?
Not yet. Maybe in the future, not to preventing, but to identify, and I think more depending on resources, in identifying subset of patients, in theory, single patient with some features, and that allow, for example, some specific diseases. So, you know, the dream of precision medicine, but at least a subset of patients with different risk score, different risk, that I think would be not impossible in the future. I don't know if in the near future but I think being rare, specifically the autoimmune liver diseases, I don't think we should move or we will have resources enough to do population screening, but maybe in pre-selected population with higher risk might be possible, not for all the population, the world population. Again, when you have diagnosis, to better characterize in order to have a more specific therapy or management, clinical management. That is my thought. But not today. and I don't know when,
We
but
must be
in
patient.
the near future.
We must be patient.
Yeah.
Okay. Any other question?
Yes.
Points?
Hi Pietro, I'm Lori.
Ciao, Lori.
What about ciao? What about relatives of these patients? Do you think we can make the same studies?
Other group, a number of groups are performing these. I think the best, what I mentioned, the polygenic risk score. So I think we should use this kind of approach that is quite cheap,
Yeah.
and might be used. We need to demonstrate that. So we need to perform a study and showing that is cost effectiveness in relatives. That kind of approach might have a real reason to be developed in the future for the clinic, in the clinic, to be used in the clinical and developed in the clinical setting. For scientific point of view, that is another story. But for the clinical setting, the use of polygenic risk score is, I think we should move on that.
Okay. Thank you.
Thank you. Any other question or comments? Well, if not, thank you very much. It's been a very informative, exciting, a good gym for the brain to follow and jump from one place to the other. And thank you very much again. If you don't mind, you can stay. I'm going to ask you something. So the other people can leave if they want. And thank you very, very much.
Bye.
Thank you.